IB Bio, Topic 11, Part 11, Sexual Reproduction C, focuses on the early moments of pregnancy, the development of the embryo into a blastocyst, and details about the placenta. The essential idea is sexual reproduction involves the development and fusion of haploid gametes. Here is the outline of all the available movies for Topic 11, Animal Physiology. Use this outline to find the movie you need for review. This movie is focused here. Remember that mammals utilize internal fertilization as an evolutionary adaptation to living on land, a difficult, dry environment. And as a result of employing internal fertilization, mammals provide high protection to the development of their young, not to mention giving birth to just a few young. When mating couples give birth to low numbers of offspring, successful reproduction requires a high investment of parental energy to ensure the survival of their mammalian offspring. Here are the first IB syllabus statements of the movie. Outline early embryo development up to the implantation of the blastocyst. State that HCG stimulates the ovary to secrete progesterone during early pregnancy. I've started the coverage of this latter statement in the last movie, Part 10. In this image, we can see the secondary oocyte with its attendant fleet of cells and a polar body. One sperm cell arrives and digests through those attendant cells, known as the corona radiata, using the acrosome reaction. You can almost see the cortical reaction beginning to occur here. The sperm enters the egg. Meiosis II is completed in the oocyte, forming a second polar body, seen here. Then the nuclei fuse, the haploid nuclei fuse, and technically this event is called fertilization. Fertilization forms the diploid zygote. Then the zygote divides mitotically, and this is called cleavage. 1 cell to 2, 2 to 4, 4 cells to 8, 8 to 16 to 32 to become a fluid-filled structure known as the blastocyst. The blastocyst implants in the uterus. In this image, we can see mitotic cleavage as the embryo makes its way down the oviduct, and then the implantation of the blastocyst in the uterine wall. In this image, following fertilization, the events of mitotic cleavage are labeled by the day. The blastocyst, a very young embryo, implants on the uterine wall approximately day 6 after fertilization. One more diagram to strengthen your understanding. Following fertilization, we can see mitosis occurring, known as cleavage. 1 cell to 2, 2 to 4, 4 to 8, 8 to 16, and so forth to form the blastocyst. The blastocyst implants on the uterine wall. Remember that tissues surrounding the developing ovary secrete HCG. HCG targets the corpus luteum within the ovary. In turn, the corpus luteum remains active through the first 3 months of pregnancy, producing progesterone that supports the development of the embryo in the uterine wall. Here are micrographs showing the 4-cell embryo on the left and the 8-cell embryo on the right. These have formed through mitosis, a process called cleavage. Here's an image showing mitosis to produce the 4-cell embryo. It's a nice image because you can see the spindle fibers formed during mitosis in a 3-dimensional representation. The morula, a day 4 embryo, is composed of 16 to 64 cells. The blastocyst is a multicellular fluid-filled structure ready for implantation. 24 hours after fertilization, mitosis has produced a ball of 8 cells. Then after 3 to 4 days, the embryo is composed of 16 to 64 cells. The blastocyst is ready to implant on the uterine wall on day 6 or 7. Here's another look at mitotic cleavage in a frog embryo. Notice that the mitotic divisions are not all equal. Some cells are smaller or larger than others. Here are micrographs showing the embryo at the 8-cell, 32-cell, and blastocyst stages. Here is a micrograph showing the implantation of the blastocyst into the endometrium of the uterine wall. Here's a diagram that might help you better understand the embryo. At the blastocyst stage, not all the cells are destined to become the fetus. Some cells of the blastocyst, seen here as the outer edge of cells known as the trophoblast, these cells will develop into the placenta and a structure called the chorion. These tissues secrete HCG. The inner cell mass becomes the fetus. Here's another diagram of the blastocyst implanting on the uterine wall. The cells of the trophoblast will become part of the placenta and chorion. These cells secrete HCG that maintains the corpus luteum for approximately 3 months of pregnancy. Once implantation occurs, development gets complex in a hurry. So complex, in fact, that I do not expect you to understand this image shown 14 days after fertilization. The only thing to note in this diagram is that the embryo is now completely embedded within the tissues of the uterine lining, and the tissues here are beginning to form the placenta, where finger-like projections known as villi provide high surface area for the exchange of materials between maternal mother's blood and the blood of the fetus. I've just covered this top IB syllabus statement. Implantation of the blastocyst in the endometrium is essential for the continuation of pregnancy. Here are 2 additional IB syllabus statements that will carry us up to the moment of birth, where we will bump into one more syllabus statement. Explain how the structure and functions of the placenta, including its hormonal role in the secretion of estrogen and progesterone, maintain pregnancy. State that materials are exchanged between the maternal and fetal blood in the placenta. In this diagram, we can see a fetus developing within the uterus. The placenta is seen here, and it includes the chorion in blue, which is a tissue layer that separates maternal blood from fetal blood. The chorion regulates the exchange of materials between the mother and the fetus. You can see the high surface area of what are called chorionic villi. The high surface area of the villi will increase the quantity of materials exchanged, such as oxygen, carbon dioxide, glucose, urea, amino acids, and water. I've provided the direction of movement of these materials. This is something to study. The amnion tissue layer in red forms the umbilical cord and encloses the fetus in a fluid-filled sac that protects the fetus. In this diagram, we can see the developing fetus within the fluid-filled amniotic sac that protects the fetus from bumps that might occur as the mother goes about daily life. The placenta is seen here with its various membranes. Maternal and fetal blood are separated by the tissue layer called the chorion. The high surface area of the chorionic villi facilitates the exchange of oxygen, carbon dioxide, glucose, urea, amino acids, and water. You should be able to determine the direction of movement of these various molecules. By the 12th week, end of 3 months, the corpus luteum degenerates, and the placenta picks up the secretion of both progesterone and estrogen. Progesterone and estrogen target the uterus to maintain the uterine lining during the remainder of the pregnancy. This is a nice diagram that shows the placenta here with maternal and fetal blood vessels near each other for the exchange of materials. To best understand what's happening, look at the arrows that show the direction of blood flow. The blue arrow in the umbilical cord shows deoxygenated blood flowing in the umbilical artery, high in carbon dioxide and urea, heading to and arriving at the placenta. The blood gives up urea and carbon dioxide to the mother, and in turn picks up oxygen, glucose, amino acids, and water, and returns to the fetus in the umbilical vein, indicated in red. In this diagram, we can see the fetus developing within the uterus. We can see the placenta here. Remember that for the first 3 months, the embryo secretes HCG that targets the corpus luteum within the ovary to maintain an active corpus luteum that in turn secretes progesterone and estrogen, and these target the uterine lining to maintain the lining during pregnancy. After 3 months, the placenta takes over the secretion of estrogen and progesterone that targets the uterine lining to sustain pregnancy. Here's the relevant IB syllabus statement. Outline the role of human chorionic gonadotropin, HCG, in early pregnancy. Here's a graph of the relative levels of hormones during the 40 weeks of pregnancy. HCG levels are high in the first 3 months to maintain the corpus luteum and the corpus luteum secretion of progesterone. And estrogen and progesterone levels increase through pregnancy as the placenta takes over the secretion of estrogen and progesterone in the last 6 months of pregnancy. Keep things straight. HCG targets the corpus luteum within the ovary, while estrogen and progesterone target the uterine lining. Here's a table with information about estrogen, progesterone, and HCG. Most of this should be familiar, but I want to introduce some detail that foreshadows birth. Notice the role of progesterone in inhibiting uterine contractions. Progesterone inhibits a hormone called oxytocin that stimulates contractions. Notice the role of estrogen in increasing the uterine sensitivity to oxytocin. You will hear more about oxytocin and uterine contractions at the end of the movie when I discuss the details of birth. Take some time to study this slide. So I've come back to this slide to remind you that the placenta has 2 big roles to play during pregnancy. First, the placenta is the exchange site for materials exchanged between the mother and the fetus. Secondly, the placenta is an endocrine gland that secretes estrogen and progesterone that sustains the pregnancy through the 9 months. When pregnancy occurs, HCG can be found in the urine of the female, and we can run pregnancy tests that test for the presence of HCG in the urine. Antibodies are constructed that bind to the HCG, and these are called monoclonal antibodies. The antibodies are attached to a stick with a color indicator. The home pregnancy test asks users to dip the stick into the urine, and if HCG binds to the antibodies on the stick, the appropriate color change occurs. Pregnant. Here is an image of the materials included in pregnancy tests. Monoclonal antibodies against HCG are attached to a stick, and these antibodies are used to determine pregnancy. If HCG is present in the urine, the stick indicates pregnancy when 2 red bands appear. With this diagram, I've come back to look more closely at the placenta. The placenta is an organ composed of both maternal and fetal tissues. Fetal blood arrives and departs the placenta in the umbilical cord. Fetal blood arrives in the blue umbilical artery and departs in the red umbilical vein. Within the placenta, the blood vessels of the fetus are separated from the blood of the mother by just a single layer of cells. There is a high surface area to facilitate the exchange of various molecules. Maternal blood bathes these open sinuses surrounding fetal blood vessels. Another image of the placenta, an organ composed of both maternal and fetal tissues. The umbilical cord brings blood to and from the placenta. Maternal blood fills the space that surrounds the fetal blood vessels. Can you list the materials that are exchanged between the mother and fetus? Can you state the direction of movement of each of the molecules? One note on the exchange of materials. Maternal antibodies can cross the placenta, giving the fetus some passive protection against pathogens, which are disease-causing organisms. Viruses can also cross the placenta. HIV-positive mothers can give birth to HIV-positive infants. Alcohol, caffeine, and other drugs will also cross the placenta to enter the blood of the fetus. Pregnant mothers need to be careful about what they ingest. In this image, we can see the 8 to 9 week old fetus surrounded by the fluid-filled amniotic sac. The fluid-filled sac protects the fetus from the bumps taken during the daily life of the mother. Here's a nice image showing the 10-week-old fetus developing in the uterus of the mother. You should be comfortable with these IB syllabus statements. Explain how the structure and functions of the placenta, including its hormonal role in the secretion of estrogen and progesterone, maintain pregnancy. State that materials are exchanged between the maternal and fetal blood in the placenta. And HCG stimulates the ovary to secrete progesterone during early pregnancy. Monoclonal antibodies to HCG are used in pregnancy tests. And that brings us to the end of IB Bio, Topic 11, Animal Physiology, Part 11, Sexual Reproduction, C. In Reproduction D, we will focus on the details surrounding birth.